Melatonin is produced in the pineal gland inside of the brain. Referred to as the sleep hormone, the brain increases its production as a result of darkness, which helps to manage the body's circadian rhythm. When melatonin is produced and travels through the bloodstream, it binds to MT1, the melatonin receptor. This is a G protein coupled receptor, meaning that it has an inactive G protein attached to it. When melatonin binds to MT1, it causes GDP to be substituted for GTP, which then activates the alpha subunit of the G protein. The alpha subunit deactivates the enzyme adenylyl cyclase, which causes a decrease in levels of CAMP. CAMP is a second messenger that usually initiates a phosphorylation cascade where protein kinases phosphorylate each other. Without melatonin preventing the creation of more CAMP, this phosphorylation cascade usually ends in a phosphorylated gene transcription factor, CREB, transcribing so-called clock genes, which create proteins that regulate and maintain neuron firing rates, keeping the person's brain activity up and thus keeping them awake. However, a decrease in CAMP levels stops this phosphorylation cascade from happening, which inhibits the gene transcription of those genes and makes the person sleepy. In addition, when the G protein is originally activated, the beta and gamma subunits of the G protein detach and activate phosphoinositide 3 kinase, an enzyme that triggers a phosphorylation cascade, specifically the MAP kinase signaling cascade. This cascade leads to the phosphorylation of a gene transcription factor, which then activates many genes related to circadian rhythm regulation.